Yeah, so I'm excited to have the opportunity to teach microbiologists about a little bit more about plant bacteriology, and I think Danny gave an excellent introduction. I loved bacteria and then kind of stumbled into this field and originally ruled out plants because I had a mentor who said that plants were boring and hard, and I had no other reason to you know, consider that. And so I kind of stumbled into this field by luck after that and became really impressed with how a lot of the same molecular mechanisms between both bacteria and their animal hosts and bacteria and their plant hosts are really conserved. But there's also a lot of parallel convergently evolved functions as far as how the eukaryotic partner interacts with plants, which Danny touched on a little bit. For today's lecture, we're going to talk about this one genus of bacteria that is Ralstonia. And I've designed the lecture to kind of go from the ecology, big picture, all the way down to a lot of the molecular strategies that Ralstonia and many bacteria use to interact with their eukaryotic hosts. A lot of these strategies individually are used by commensals and mutualists, but Ralstonia is kind of a multi-tool pathogen and has like the whole kit and caboodle and causes a particularly aggressive plant disease. When we get into the traits section, we're talking about a number of different molecular strategies, some of which you have learned about in this quarter. And I'd like you to, I've left it blank, and I would like you to write down what is the purpose of these tools? How do they help the bacteria grow, survive, and cause disease in plants? So Ralstonia solanacearum causes a disease called bacterial wilt disease. Plant pathogens are a little bit different than animal pathogens in one sense. All plants are cold-blooded. They don't have, or not really blooded at all, but they're not warm-blooded. So because of that, the microbes have to be adapted to the local weather conditions. There's some pathogens that prefer cool climates and some that prefer warm tropical climates. Another reason why pathogens have such a tie to the environmental conditions and the weather is that plants don't move individually through the department. They can they don't move through the environment. They can disperse with the seed, but an individual plant roots and pretty much stays put. So unlike animals, which can intermingle, breathe on each other, and spread pathogens, plant pathogens must have their own mechanisms to transmit themselves between plant hosts. Some of them can use insect vectors, the plant equivalent of mosquitoes, but a lot of them have to transmit through the wind, the rain, the soil, and so forth. So in the case of Ralstonia, this is a pathogen that prefers warm temperatures. And since it's a bacteria that doesn't survive drying out well, and it's transmitted through the soil, it requires year-round precipitation so that the soil doesn't dry out too much. So fingers crossed that California doesn't get Ralstonia. In the US, Ralstonia is endemic in the Southeast US, Puerto Rico, Hawaii, and Guam. And it's pretty ubiquitously widespread in the tropics around the world. There's a few major plants that are hosts for Ralstonia. Two of them that I'm highlighting here are in the Solanaceae botanical family, which is what gives this pathogen its very tongue twister species name, Solanacearum. So one of those is tomato. Tomato is probably one of the most susceptible crops. Like many of these different diverse lineages of Ralstonia all can unfortunately wipe out tomatoes. And they're an important food crop, an important economic vehicle, especially for small growers, because they can make a lot of money selling them in the market. But potato is probably one of the biggest impacts that Ralstonia has in terms of global health. Because potato itself is a very important crop around the world, is one of the few nutritionally complete plant products. So it has the complete essential vitamins and amino acids. So, that's why, unfortunately, in Ireland, there's Irish potato famine, but there was a long-standing time where that was the one and only source of nutrients, and people were able to survive eating just potatoes. But it's a really hardy crop. You can see that Ralstonia is actually infecting this potato plant. It's colonizing the vascular ring here, and there's white bacterial goo. And it's surrounded by necrotic rot. And so those potatoes won't be edible. And even worse, they have the pathogen, and so they can transmit the pathogen to a new field, because potato tubers are cut up and used as the seed. It's called seed. It's not a true seed. They're vegetatively propagated, and the next plant cycle comes from the potato tubers. Banana is also a really important crop that Ralstonia infects. It causes a lot of issues in South America. Luckily, Ralstonia is not actually in Africa, or at least the banana strains are not in Africa. And there's another strain in Indonesia that causes what's called banana blood disease, because when it colonizes the vascular ring of the bananas, it makes the banana produce a red pigment that kind of looks like blood, and the bananas are inedible. And so we're familiar with dessert bananas. Those are exported from the tropics, and we eat a lot of those. But around the world, there's a lot of starchy and dessert bananas that are used as a large part of local cuisines. So Ralstonia's infection of those bananas is not good. Okay. So now to move into the life cycle and think about the ecology within, the plant soil cycle. Ralstonia starts by coming from the soil and invading roots. It then gets into the xylem vessels, which Danny mentioned are able to transport water. So that actually is vaguely similar to our blood. There's a lot of differences, but it is a vascular system that transports liquid through the plant. Ralstonia will then spread systemically through those xylem vessels. It can bust out of them and grow in the intracellular spaces in the stem. And by clogging the vessels and causing this damage, it prevents the plant from taking up sufficient water and causes wilt, which is sometimes I refer to it as the plant version of a heart attack, because it is, the clogging of the vascular system. Throughout infection, the bacteria will also shed from roots and get back out into the soil, where they can be washed down a hill through irrigation water or precipitation. They can also enter irrigation creeks and irrigation ponds, where they can then be used and sprayed onto the next crop, and that can allow them to transmit locally. But not particularly long distances. For the long distance movement of this pathogen and a good number of pathogens, one of the biggest problems are plants that are latently infected, and then humans move them to a new region. So of course there are systems in place and regulations to try to prevent this, but some slip through. And the latently, the fact that especially crops like potato and banana are vegetatively propagated, a lot of pathogens can potentially cause latent infections that you don't see, and then they can be moved into that new location. And generally for transmission, Ralstonia, is kind of on its own, and as long as it doesn't dry out, it's good to move. Ornamental plants are like kind of the plant version of zoonoses. A lot of them are propagated in the tropics and then exported around the world. And so that's a major vehicle where plant pathogens can be moved into new locations. There are some regulations and people do, try to follow best practices to limit this, but they are definitely less regulated than food crops. And so they can be a major way that pathogens can come in, and then they might be able to infect current crops. I think that's what happened with the citrus industry in Florida. So you should read news articles about that. I like to think about the evolutionary neighborhood of my bacteria. So Ralstonia is a beta proteobacteria. You might have heard of Neisseria gonorrhea, Burkholderia, probably more Neisseria. But I think some of these are ones that you've heard this quarter, but don't need to dwell on that. So now we're in the meat and potatoes, and we're going to be focusing on some different traits that Ralstonia has that allow it to overcome its plant hosts, and many other mutualists, commensals, and other pathogens of all kinds of eukaryotes use a lot of these strategies. So one of them is that Ralstonia uses chemotaxis to sense the chemicals that plant roots exude. So they exude a lot of organic acids and things that Ralstonia can sniff out with its chemoreceptors. And then it directs its swimming motility to bring it towards that higher concentration gradient where the root is. In addition to some of those nutrient sources that are exuded from roots, roots of plants also make a large amount of these phenolic compounds that are antimicrobial compounds. So you can probably recognize those phenolic structures from OCHEM. And this is one class of these plant phenolics that are called hydroxycinnamic acids. Ralstonia has a pathway that allows it to break these compounds all the way down into carbon sources. And that provides two functions for Ralstonia. One, phenolic compounds are a little bit, toxic. They can cause some oxidative stress and damage lipids. And then also it provides a minor amount of food. So it's, taking a bad thing and putting two wins in there for the bug. And in my PhD, I actually worked on this pathway and found that Ralstonia needs it to colonize roots, but at later stages of disease, it no longer needs this trait. So it's an early trait. This is one of the coolest traits that Ralstonia has, and it was discovered in part by my lab mate, Tuan Tran. So it starts with knowing about this very interesting cell type on plant roots that are called root border cells. And these are produced at the tip of a root, and they actually fall off of the root as it grows, and they become these little free living, cells sitting in the soil. And they just kind of sit there until they sense a pathogen through some of those similar, well, it's not worked out in detail, but probably through a lot of those receptors that Danny talked about. And when they sense a pathogen, what they do is they take their nucleus and they kind of open it up and throw their chromatin, their DNA, their chromosomes out of their cell. And it makes this like gooey mesh of a gel that we call a net, or a nucleic acid extracellular trap. And that's able to actually catch Ralstonia and some other pathogens. So so not only does it trap them and kind of hold them immobilized, but Histones, the structural proteins of chromatin, are positively charged proteins, and pretty much all positively charged proteins turn out to be quite toxic to gram-negative bacteria. They disrupt the negative charge of the outer membrane, and so histones can actually kill bacteria. And so this is like such a cool evolutionary thing where plants have evolved to use their DNA and their histones to be an immune defense. And what's even neater is that this is a convergent evolution. In animal immunity, neutrophil cells can also make a chromatin trap like this. So it's really neat. And so Ralstonia, and then of course some animal pathogens, often have these DNases that can degrade the DNA that they secrete. So that helps them free themselves. Okay. So now moving away from roots and thinking about Ralstonia, especially in the xylem, where it's starting to get high density, really clog up those vessels. So like many pathogens, Ralstonia has this trait called quorum sensing, and that allows it to coordinate a lot of its behaviors based on its cell density. And so quorum sensing works because bacteria produce and secrete some unique secondary molecules, and they, secrete them outside themselves. When Ralstonia is at low cell density, like when it's in the soil and it's away from a plant, that signal, it produces it, but then it kind of diffuses away and it stays at really low concentration. In contrast, when they start to become very dense, like when they're inside the plant, you have a whole bunch of those same cells that are all producing that same molecule, and the concentration of it rises. and it hits a threshold concentration where now the physical properties of, chemistry allow it to bind the receptor on the bacterial membrane that senses the signal. And then you get a whole bunch of phosphorylation cascades, a lot of cell signaling, and you get a change program of gene expression, which then impacts specific physiology functions of the cell. And you can also think of that as changing the bacterial's behavior from a free living to a virulent phase. One underappreciated thing that is often regulated with quorum sensing are metabolic strategies. So this is a metabolic map of some of Ralstonia's catabolism. So it can eat a whole bunch of different carbon sources. And quorum sensing controls a shift between being a generalist to being a specific, preferencing some specific compounds. So when it's in the soil, it kind of wants to be ready for any carbon that comes around so it can take advantage of it. And then at high cell density, which is when it's in the plant, it shifts to preferring, in green here, some of these sugars, including sucrose, which is a major sugar in plants. And so quorum sensing, shifts the metabolism, but it also turns on these other traits here. So extracellular polysaccharides. Most pathogens make an extracellular polysaccharide. It can be free, it can be capsule and very tightly attached to cells. And these EPSs generally protect bacteria because they form a hydrogel. And that gel is viscous. It slows the diffusion of toxic chemicals to keep them from hitting the bacteria as often. These sugars can also absorb water and keep some water near the bacteria, which protects them from some desiccation stress. And they can also protect the bacteria from the entry of phages and other sorts of intermicrobial, predators happening at that scale. And in the case of Ralstonia, there is this really drippy EPS. It's got a unique, whole polysaccharide structure that I haven't seen with other bacteria, but it's probably not completely unique. It's a lot of like N-glycosylated sugars. But the physical properties of it are kind of exquisite. It's not sticky. It actually is very drippy. So in microbiology lab, you'll learn to keep your agar plate so that the agar's on top and then the lid's on the bottom, and that keeps the moisture on the lid and keeps it from washing your cells all over your plate. But if you do that with Ralstonia, the colonies will actually drip onto the lid, and it's so gross and awesome. And whenever I see a new microbiologist and I'm in lab, I make sure they see this bacteria because it's gross. And that really drippy nature is probably useful for this bug because it is a bacteria that needs to spread systemically. So it wants to have an EPS to protect itself against plant antimicrobial responses, but it doesn't want to have such a thick EPS that it gets stuck in one place. Ralstonia also has some unique respiration strategies. I guess they're not that unique, but Ralstonia, as opposed to us, which we have to breathe oxygen, Ralstonia also prefers oxygen. However, we find during infection, it prefers it so much and grows so rapidly that it really decreases the amount of oxygen that's present in the plant. And so it has to have, it's a facultative anaerobe, and it can also breathe nitrate and use nitrate as its electron acceptor. And so it's able to convert nitrate all the way down to dinitrogen gas and use this to power ATP generation. Ralstonia also needs some strategies to help it move systemically. So here's a way that the xylem is very different from our blood vessels. The xylem is more of a network of individual vessel elements, which are represented by these black lines. And they have finite ends. However, they are interconnected by semi-permeable membranes between adjacent vessels. And these membranes have pores that allow water to move through them and in the net direction of roots to leaves. But the pores are small and bacteria cannot fit through these semi-permeable membranes. But these membranes are made out of cellulose and pectin. And so a lot of xylem pathogens, and honestly plant pathogens in general, have secreted enzymes that degrade cellulose. So those are called cellulases. And they also have pectinases. But in Ralstonia's case, the cellulases are so much more important. And they really help it be able to move from vessel to vessel and break out. Ralstonia also has this really cool molecular machine called a type 6 secretion system. This is a fairly recent secretion system, in that it was discovered a decade ago as opposed to like 30 years, 40 years. And this is a multi-protein machine that acts like a spear. With this secretion system, bacteria, gram negatives, I think, are the only ones that have this, are able to load effector proteins onto the tip of the spear and then stab them into a neighboring cell and deposit them. And these effectors that are delivered through this system tend to be very violent enzymes, toxic enzymes that degrade key processes in cells. So they're often really active DNases, lipases that degrade lipids, amidases that degrade amide bonds and peptidoglycan. And they really target these central processes, degrade them, and kill their neighboring bacteria. So that allows bacteria to kind of advance their niche as they're colonizing and defending their space. But of course, when bacteria grow, they grow clonally, and their kin, their daughter cells, are right next to them. And so, evolution would not allow them to, probably. It'd be really hard to evolve a mechanism to kill your neighbor without being able to protect yourself, because you want to make sure that your daughter doesn't kill you. And so for every effector that's present in the type 6 secretion system, there's a corresponding antitoxin that's called an immunity protein. And these bind to the effectors and keep them inactive when they're inside the producing cell, or if they get injected into a kin cell. And so there's that self-protection thing. You'll learn a lot about these toxins, antitoxins, and a lot of plasmids and phages, and it's cool evolution. Okay, so now I want to just highlight a few traits that are not really actively regulated by quorum sensing, but are still essential for the pathogen to complete its ability to grow in planta. One of those really important traits is the type 4 pilus. And this is a machine that mediates what's called twitching motility, which allows bacteria to move along a surface. And it's called twitching because under a microscope, it goes back and forth from either direction. So they called it twitching because there's like these little cells going back and forth. And the way it works is this type 4 pilus extends out of the pole, it hits the surface, and then it binds. There's a conformational change, and that triggers it to retract and pull into the cell. And so when that retracts, it then drags the cell forward. And Ralstonia does not swim. It doesn't use flagellar motility when it's in the plant. It exclusively uses this type 4 pilus twitching motility to move along the xylem walls and probably like lets go sometimes and like floats like a raft in the center of the vessel. So those are the two major ways that it disperses in the plant. But the type 4 pilus has a few roles for bacteria. So in addition to doing the surface motility, that's just a colony edge. So there's some bacteria moving through the pit membranes using their pili. But there's also this second benefit that the type 4 pilus gives to bacteria. And that is that it allows them to be naturally competent and take up DNA from their environment. And so this is a labeled cell. The pilus and the cell are labeled with green. I think it was green fluorescent amino acids. And you'll see some red DNA at the end here. And let's see. I don't know. I have to apparently turn off the laser pointer to play videos. Not very used to having videos and things. There we go. So if you cut that, there's like this little red thing that gets sucked into the cell. And so that's actually a bacteria caught live on camera taking up DNA from its environment. And we had never seen that until 2018. So you should look at this blog. So that's very cutting edge and cool. And that's not a video of Ralstonia, but Ralstonia does this too. So plants are good chemists. They make a lot of antimicrobial compounds. So pretty much every plant pathogen has a number of drug efflux pumps that allow them to move those chemicals back outside of their cytosol so that, you know, they're less of a problem in the front yard than, you know, running around the house making damage. So drug efflux pumps are important. You usually hear about these in animal pathogens, especially in the context of antibiotic resistance. But you know, antibiotics are less used in treating plant diseases. Danny mentioned reactive oxygen species are a key output of plant immunity. You know, very radical, can damage a lot of bacterial cells. So a lot of pathogens have mechanisms to protect themselves from that, like catalase, which breaks down hydrogen peroxide. And then for the final trait that we're going to talk about the type 3 secretion system. So this is another very cool secretion system. You know, don't get it confused with type 6. This is type 3. It's older. People knew about it for much longer. And this one is also a multi-protein system. From a rough glance, they look a little bit similar. But whereas the type 6 acts like a spear and kind of just like stabs its effectors into the neighboring cells, this is a much more discrete secretion system in every way. So it still forms, and then it makes this pore at the end, and it almost exclusively, there might be there's always exceptions in biology, but for the most part, this is a system that allows bacteria to interact with eukaryotic cells. They interact so there's a certain type that's longer for plant pathogens because it has to be able to extend past that plant cell wall to the membrane of the plant cell. But there's parallel secretion systems between animal and plant pathogens. And this one is a hypodermic needle where the effectors are actually pushed through the center of the pilus and then deposited into the plant cytosol where they can have their activity. And these effectors, and that's just a jargon for they have an effect. Like it's now a noun of it has an effect. But it's a way to make it a little bit noun where, you know, we don't know what they do, but they have an effect. Type 3 effectors are kind of stealthy spy tools for pathogens. Instead of being brute force weapons, they very rarely are necrotic or kill the host cell. They're usually pulling tiny strings to manipulate host cell biology to benefit the microbe. And so, of course, your pathogen, or there's also the system used by rhizobia that forms nitrogen fixation nodules. That's a mutualist of plants. So this is used not just, you know, it's been used by really strong mutualists and pathogens. And the key thing is that it allows the bacteria to interact with the eukaryotic cell and change the eukaryotic cell biology to benefit the bacteria. And so, you know, key things there are increasing nutrient transport to where the pathogen is, shifting metabolism to, you know, help promote the pathogen growth. And then also a lot of these effectors can also suppress plant immunity. Some of the effectors are kinases or phosphatases that change the phosphorylation cascades. Some are proteases that break down a certain, you know, key protein. One really cool type of effector are they actually translocate into the nucleus of plant cells, and then they they have domains in them that mimic transcription factors of eukaryotic cells. So they're able to bind to the DNA of a eukaryote, and they have a specific region. And they can actually activate certain promoters and manipulate gene expression of the plant. And some of those actually turn on sugar transporter genes called sweet genes. And so it's a really cool way that the bacteria gets the plant to give it nutrients. And so not every gram-negative bacteria, but a lot of the gram-negative plant pathogens have this type 3 secretion system. I'm skipping this. So a lot of them have that core system, but then the specific, toolkit that they have, their repertoire of effectors is very variable between strains. And that's because there's this coevolution, Red Queen hypothesis sort of model of coevolution between a predator and a prey that drives, molecular innovations. So pathogens have this, evolutionary desire to come up with new ways to manipulate their plant host, and maybe they get those through horizontal gene transfer. But the plants can often use those NLR receptors that Danny talked about to bind to effectors or to look out for the damage, the subtle damage caused by those effectors. And so there's this arms race that drives some of the variation within the whole Ralstonia genus or pathogen genus that you're looking at. And so this table, Ralstonia has tons of effectors. Like, it's probably the most prolific for the plant pathogen, 60 to 80, which is a lot. So that's any given strain. And then there's a lot of variation between them. So some strains are missing some, and some strains have, the effector. And it's probably this evolutionary history of which host plants the strain was adapting to at the time that drives the pathogen's repertoire. I didn't really touch on soil and water survival traits because we don't actually know much about those. But that's actually a research goal of my lab, is to try to understand some of this, some of the ways that Ralstonia survives in the environment and also interacts with the microbiome when it's outside.